Hey everybody! Today we're going to be showing you a video on how to do a sickle cell screen. This will hopefully help us go a little bit quicker in class since you've already seen it and you can get moving on it right away. Um, Basically, a sickle cell screen is going to help determine whether you have a hemoglobin S in your blood. So whether that be hemoglobin S trait, meaning a heterozygous, or a hemoglobin S disease or sickle cell disease, meaning a homozygous S. And this cannot differentiate between the two, so if you do get a positive, you'd want to confirm it with hemoglobin electrophoresis. And how this works is we're going to be adding this phosphate buffer to these tubes, and then we're going to be adding a little bit of blood to that tube, and the phosphate buffer is going to lyse the RBCs, and the hemoglobin will then be released from the red blood cells. And pretty much all hemoglobins are going to be soluble in the solution. So you'll be able to basically see right through that tube. It may be a little bit red, but you can see through it. It's clear. Where if you have hemoglobin S in your blood, it basically is going to form little crystals. That hemoglobin S is not soluble in this phosphate buffer, and if you have all those little crystals forming, it's going to make the solution look very turbid. So let's start the test. How do we do this? So this test is going to require each tube to have 2 mLs of this phosphate buffer. This is already pre-made up. And you're always going to want to run your QC. So we have a positive QC, and we also have our negative QC. Yep. And then we have our patient. So we always want to label our tubes, and I'm on an angle here, so please forgive me. So positive, negative, that's our QC, and then our patient, we'll call him Bob. Bob. We'll call him Bob. So Bob is going to get screened for sickle cell today. Now we can measure this out, or we have this handy dandy card. Thank you to whatever student left this out because I couldn't find a proper rack in here. I'm sure that in this lab there is hemoglobin sickle cell screening racks, but this one will do for our video. And if we put this card right behind it, that black line is our 2 ml line when it's measured out on these tubes. So I can simply just kind of hold that there and hopefully without spilling, and I'm on an angle here, fill these tubes up to about that black line with our phosphate buffer. It doesn't have to be exact, but you want to be pretty close. Whoops, don't do that. Then this card can go away now for a second. That was just to help me get my 2 mLs. And so now we need to add our sample. We always want to mix up our samples with this QC. You're going to want to roll it in between your hands for about 10 seconds. I've already done this, so I'm going to go a little bit quicker. You're going to flip it upside down, and you're going to roll it in your hands. And then you're going to want to invert it 6 to 8 times just to get it all suspended correctly. When we're adding these to the tubes, I like to start with my patient first, and then I like to do my negative QC, and then I like to put my positive QC. Is it mandatory that you do that? No. But if there is any cross-contamination, I don't want any of that going into the patient's tubes. So I'm going to start with that one first. This test for this patient requires 20 microliters. So we have our pipette here, and I'm going to pipe out 20 microliters. And then after I pipe out 20 microliters, I'm going to want to cover this and shake it up. And there's many ways to shake it up. You can use parafilm. You could use a glove if you changed it. I like to use these orange pads. One side is got that orange, and it's I don't know what you call it, like plasticky almost. The other side is absorbent. You don't want to use the absorbent side because your specimen could leak. So I just put it right on the top there, and then I shake it up. And then I'm going to want to throw that away. And I'll put this in my tube. So that's our patient. And then our QCs are going to require 1 drop. And then I like to go to the negative QC next. And we're going to do the same thing. And if I was accidentally to cross-contaminate, it's the negative QC. So that's good. And we'll shake it up again. We'll put that in our rack. And again, I'm throwing that away. And then we have our positive QC. And that's also 1 drop. Sometimes these QC vials can get a little messy. And we'll shake that one up. Alright, so when we're looking at these, we're going to put our card behind it. We're going to set a timer for about 10 minutes, and then we'll come back and we'll see the results. Okay, our time is up, our 10 minutes is up. So what we're going to do is look at the solubility, hard to say that sometimes, of our cards. So when we're looking at this, we'll start here with our positive control. And I'm looking to see if I can see the lines of the index card behind it. And remember that hemoglobin S is not soluble in this phosphate buffer. So it's forming all those crystals. So that's why we cannot see through this tube. So this is considered a passing control for our positive. Now we look at our negative. So there should not be any hemoglobin S in this, and as you can see, we can see the lines of the index card behind or through this phosphate buffer. And we can see Bob's sample, our patient, who has been screened for sickle cell. You can also see through this solution, and you can see the lines of the index card behind it. So we know Bob does not have S hemoglobin. So he does not have the hemoglobin S trait, and he does not have hemoglobin S disease. Let me know if you have any questions. This is the sickle cell screen.